Hello, everybody. We're just about ready to get started now. So I'd like to welcome you to the webinar for getting started with image cytometry. My name is Jamie McCarthy, and I'm a technical application specialist with de novo software. I'm really excited to be hosting this webinar for you today, so I can show you some of the things FCS Express can offer for our users. So I'm going to begin with a little bit of background information about our company. De novo software has been providing cytometry analysis software for nearly 25 years now. Our headquarters are based in Pasadena, California, but we have application specialists and distributors located around the world. This enables us to make our customer support really exceptional. We really want you to be getting the most out of the use of FCS Express. So if you ever write into support with questions, we'll try to answer you with a few hours and never more than 24 hours. And we're able to do this because of the way that we have people look in different locations. For example, I'm over in the UK, so if you ever are local and write in a question, we're able to get back to you a little bit quicker. We also offer free one-on-one or group training sessions that you can bring your own data with, and you can start analyzing and using FCS Express in exactly the way that you want to. So FCS Express is not only for analyzing flow cytometry data, but it can also provide you the tools you need to analyze your image cytometry data as well. Today's webinar is going to introduce you to some of those tools, features, and capabilities that the software has to offer. And FCS Express has included image cytometry analysis tools beginning back in version 4, and we've included it in every version of FCS Express since then. We work closely with vendors to make our software compatible with all of the latest image file types and using all of the data from all of the latest imaging instruments. We're always trying to make improvements to our analysis tools for image cytometry data, and I'll give you a little bit of information later on how you can feedback and help us with that as well. So let's go ahead and start looking at the workflow to get image cytometry analysis started. So here's the kind of outline of our talk for today. I'm going to go over some of the topics that we're discussing today. This is not an exhaustive list. We'll kind of bounce around topics and look at a few different things as we go through our data. We'll talk about the kind of workflow to image data generation. These are things that you need to do before you come to FCS Express. This is things that you're doing kind of outside of our software. And then the rest of the time we're going to spend in FCS Express, spend the rest of the time in the software. And I'll show you the kind of FCS Express workplace interface, how to insert picture plots and scatter plots, how to use the data list to kind of organize your data, how to kind of gate and using the gating tool view to keep a track of all your gating and all your gating hierarchies as you make different populations in FCS Express. We're going to have a little look at statistics and have a look at some data grids and how to organize text tables to kind of make a really nice layout for your analysis. We're also going to be making some histograms and some overlays to compare different samples, and also a little bit of batch processing to understand how you can kind of get data out of FCS Express if you need a little summary for yourself. So there are things you're going to need to do before you can work with image data in FCS Express. The first thing you're going to need to do is you're going to need to generate an image. There are essentially 3 phases to getting image data so that it can be analyzed in FCS Express. First, you're going to need to acquire those images. You could do that on a microscope, on an imaging flow cytometer, or whatever platform you're using. The next thing you're going to need to do is you're going to need to segment those images using an image segmentation software. This is a separate software from FCS Express that does things like define cell boundaries and objects of interest. It's typically the stage where you would identify colonies, clusters, or cells. And finally, once you've kind of segmented these images and defined your objects of interest, you can then extract features such as intensities and areas. And these can be quite a lot of features and parameters. But FCS Express can handle as many features and parameters as your imaging software can produce. And you can bring all of this multi-parametric features into our software for analysis. Once you've kind of acquired your images, segmented them, extracted all the features, all you need to do is then export your data in a compatible file type. We've listed those in this table here. FCS Express can handle a wide variety of image file types. There is no universally adopted file format for image cytometry or image analysis in general. For example, there's nothing that's really a parallel to the FCS file format. The most common and probably closest to the FCS file format is probably the ICE format. ICE stands for Image Cytometry Experiment. And it's kind of the one which ISAC used to kind of keep track and try and standardize across the industry. There are a number of instruments that use the ICE format listed here. You have the CountStar, you have Nexalom, you have a phase holographic imaging, you have Thermo Scientific using the HCS Studio, Tissue-Gnostics, and Yokogawa. The software can also natively read DAF files from the Amnis Ideas instrument, NX, which we can see here, NX DAF files from the Nexalom instruments, or files from Cell Profiler or ImageJ. Cell Profiler and ImageJ are kind of third-party softwares which you can use to segment and extract any features. And so you can use those and then bring the outputs, the Cell Profiler outputs or the ImageJ outputs directly into FCS Express. If you don't see your instrument here, we're always trying to add new file types for compatibility. So I'm going to show you in a little while where you can check on our website to see the most up-to-date version of this table. And if there is a product or a file format that you wish was on here, please get in touch and we can tell you kind of if it's in the works or if we have an alternate solution for you to be getting your analysis done in FCS Express. So why use flow cytometry analysis software to analyze image data? The main principles of flow cytometry and image cytometry analysis overlap. They both take multi-channel fluorescent data and use scatter plots and histograms to look for patterns or to look for statistically significant differences. And once the data has been analyzed, once you've done some image cytometry analysis, you want to make it easy to share your results. And we think that a tool like FCS Express can really help you in this aspect. The first step of any FCS Express analysis is to build a layout. You can add plots and gates to your layout and include statistics tables and graphs to show your results. Once you've built a kind of analysis strategy up, you can display original images to kind of confirm your statistics or to help tell your story. And you'll see the actual images of your cells with all the different gates potentially showing up on your picture plots. Finally, when the layout and analysis is complete, you can generate your reports. These can be kind of presentation-ready reports with high-resolution images that are straight ready for publication. You can export into PowerPoint, PDF, or Excel, depending on what output you're looking for. And in this webinar, I'm going to show you exactly how easy it is to build this layout and turn it into a publication-ready result. So you may have noticed that I'm already presenting to you directly from FCS Express. And in this way, I'm trying to show you that you can make these kind of really nice final results, these kind of presentation-ready results. We're able to use all of these other features to tell a story and to have a nice kind of final result for us. But I'm going to take a step back and show you what happens when you first open FCS Express. You get this kind of a launch screen here, and there's a few things here which I think are really useful if you're just starting up an FCS Express that I want to show you today. The first is going to be a lot of these are in this kind of center section here, and what I'm going to do is I'm going to open up this videos tab from the launch screen. And what this does is it brings us to this little collection of kind of videos and webinars, and these are kind of organized by subject. So if there's this particular thing you're wanting to learn and you want to see, do we have a video for that? You can come in here. For example, we can come in here to image and we can find all the videos that we have as imaging, such as this webinar will be in here. And also we have a few different videos about kind of Amnis or about pipelines in FCS Express. This is where you can do things like the unsupervised dimensionality reduction and clustering and applied into images can be a really nice tool to use. But of course, you can come back and look at any kind of different. If you're not looking to learn about imaging that day, you can of course come in here and learn a different topic. The other section, which I think is really nice, if you prefer to learn from kind of articles rather than videos, you prefer kind of written instructions, you can come to this knowledge base. And this is once again just an organization of different articles put together by topic. So you can come in here. The one that I want to point you in the direction of is in image cytometry. In this other image cytometry, we have this file formats article. And I'm opening this up because it's the home for that table that I showed you in the presentation. So you don't need to watch back this presentation to find this table again. You can find it on our website. And it's also where we're updating this table. So as we add new products or new file formats, we'll put them in here. For example, most recently, we've added the attuned SciPix from Thermo Scientific. And you can see, that you can bring the ACS or the FCS files in from that software. The last tool from this launch screen, which I think is really useful, is the helper manual. In here, you can come in here and, this is a little bit like the Bible of FCS Express. You can come in here and you can find any kind of particular thing that you're looking for. You can come in here and find, the different plot types, and, you can have a look at how best to change all of those. We also have a tutorial at the end of this manual, and we even have an introduction to FCS Express Image Cytometry portion of the tutorial. And you can download some example data, download some example layouts, and kind of have a practice working through the type of analysis that you can do with us. So those are kind of 3 places that you can go for help whilst you're learning FCS Express, or if you've got a trouble and you want to try and solve a little problem, those are the 3 places that I'd recommend looking. If you are brand new to FCS Express, on the right, you're probably not going to have any of these previously layouts for you to open, but what you can do is you can come to new layout. And you can see this is what FCS Express looks like. It does look a little bit like PowerPoint, and that's definitely by design, because it means that you already are kind of 50% to the way to knowing already how to use FCS Express. For example, I know if I go to File, it's where I can open layouts, save layouts, also have a little bit of kind of administration as well in here as well. And as I go along these tabs, everything that I want to do is found somewhere in here. If I want to add a new page, I can find it in the Home tab. If I have multiple objects and I want to start arranging or aligning them because I want to be building up a nice, a nice layout and a nice kind of final result, I can do that in here. If I want to insert an object, I come to Insert. It's where I can insert some of the things we'd expect, like our kind of 2D plots, but also the picture plot, and any other objects which are useful for building out that nice layout, like a picture or a text box, or if we're going to be doing some spreadsheets later. Design is where I can flip this over to landscape because I think it presents a little bit better for examples like today. And if I want to start inserting a gate, I come to the gating tab. If I want to start exporting some of my data, I can come to the batch tab, and I can, we're going to go over batch analysis a little bit later. If you're coming back to FCS Express after a little while, you may notice we have these 2 kind of boxes already docked, Data List and Gate Navigator. They were always available in FCS Express. For example, Data List was found in Batch Data List, and the Gate Navigator was found in View Gate Navigator. But we open these by default now because we think that they're super useful for people when they're just opening their analysis. Of course, if you don't like them, you can get rid of them. But the Data List is used for kind of adding your data, organizing your data, and seeing all of the files that you have in your layout at that time. And Gate Navigator is for kind of keeping track of your gates, keeping track of the hierarchy and a few different actions that you want to do with your gates. So let's start by adding some data. So what I would do is I'd go to the data list and click this little plus. This then takes me to this little box, which is this little window, which is going to help me, and I can go and find some data. So let's find some images for us today. Now a note, if you are in a folder expecting to see some data and you can't, it may be because of this little drop-down menu here. We mentioned all of the different file types that you can have with image cytometry. And what that means is you kind of have to be a little bit intentional sometimes if you're looking for a particular file type. For example, I'm looking at some cell profiler data here, so I have the cell profiler output selected. But if I was, previously been looking at some FCS files and I came looking for my cell profiler data, I wouldn't be able to see it. So if you ever can't see some data, don't feel like that they're not available for you to bring in. It could just be that you have this selection in here. A little trick can be using this FCS files with the little dot in it, and that kind of shows you all files. But for today, I just want to bring in this cell profiler data. So let's do that. This is now in my data list. And what I can do is I can rearrange them if I wanted to, using the little up and down arrows. If there's a particular order that I want to be looking at my data in, I could also go and find some more data again, open up this and go searching for some more data. I can also remove some data if I wanted to, using this little delete button. Say I brought a file in I didn't want to be seeing anymore. Also, if I wanted to be bringing in files from multiple folders that are all kind of contained in one big kind of overview folder, I can just use this search function, search all the folders inside and bring in just the data I want. If I want to go and start with adding some data, I can go to insert and add the picture plot. But what I can also do, which I find a lot easier, is just to drag and drop over here. And then what it's going to do, it's going to ask me, okay, you want to add this file to your layout. What type of plot do you want? You have a little bit of options here. All the options that are able to you are here. For example, we could be adding, density plots or dot plots, but I want to start with a picture plot. This has now added a picture plot to my layout, and it's already kind of building up my final results, right? This is an object that I can move around, I can resize. I could also, if I wanted to, this file has two versions of the image, a cytoplasm and a DNA version. So what we can do is we can kind of flick between those on this axis here. A note about picture plots in FCS Express. They have been made to keep the resolution that the initial image was taken at, and we always start in the top left hand corner of the image. So what do I mean by that? I mean that we're currently not seeing all of the image in this picture plot. You can tell that because if I resize this to be bigger, I've kept this kind of image size, but I've just exposed a little bit more of it. Now, you may want to actually have the image zoomed out a little bit and show it all in this kind of size here, and you can do that. And this is a little bit of a theme that's going to be coming up today. If you want to change how an object behaves or looks, you go to Format. So what I can do here is I can come to Format, I can go to specific options, and now I can interact with this plot. You see this little red window here is showing me what bit of the image I'm showing. So I can move this around, I can see, wherever of the image I want to be. I can also, if I want to, zoom out. And I can resize this if I feel like I'm missing a little bit of the image or anything like that. I can do that. And now maybe I'm a little bit happier with how this image is looking. So I can close this formatting. Now we've got this kind of image kind of zoomed out and looking how we want, we can start to interact with it a bit. Maybe I want to select some of these cells specifically and kind of know a little bit more about them. I could go to gating. We have all of these gating options, to draw a round gate or a rectangle gate. We can also draw a polygon gate. These are these kind of dot-to-dot gates where you select all of the vertices, close it up, and then you get this option. This is kind of looking at the focal view, so I'm just going to call it a little FOV gate. The gate color is really useful for kind of backgating and understanding different populations, so you can kind of specify that as well. Now we've got this gate in here, you'll see that we actually have started to populate our gate navigator as well. So we had this no gate, which is basically all events or all cells, and then we know FOV gate is below that in the hierarchy. I can update this, move this around, and it's going to update this little statistic for me. Maybe I want to have a look at the DNA of the same gate position. So what I can do is I can duplicate this plot, bring this in alongside, and change this to DNA. And you'll see we're showing all of those cells in red. And as I move this around, it's going to live update that. So you see, as I'm moving this gate, it's moving those for me. But maybe I want the DNA image to live a little bit separately. Maybe I don't want to show up this gate. What do? I go to Format, and I go to Gates to Display. If I want to change something, I go to Format. Let's go to Gates to Display. And what I'm going to do is, rather than showing all gates, maybe I want to show no gates. So now I'm not going to be showing you where this FOV gate is on this plot. Or if there was a subset or a specific gate that I wanted to do, I could go to this only the gate show below. But for now, let's keep the all gate in there. If I wanted to drill down to look at just the cells in here, what I can do is I can drag and drop. And now what I've done is I have selected just those cells in the FOV gate. I've just got those in this image. This updates live. So as I move those around, just those cells are being shown. I, of course, could come in here, and if I wanted to, format this image to, maybe zoom in a little bit on those cells. Just, if I was looking for something, if I wanted to, see them in a different light, I could do. But we mentioned earlier that there's a lot of kind of carryover and a lot of similarity between image cytometry and conventional cytometry. So I can also come in here and insert some dot plots. And then all of the features that have been extracted are available for me in this dot plot. I could change those into here. So maybe I want to have a look at the intensity of the cytoplasm against this shape area parameter. A little theme that you might see with image cytometry is, because you're dealing with less events than you're used to with maybe something like flow cytometry, some of these dots might appear a bit small, or you want to see them a little bit clearer. So what you can do is you can come in here, Format. We go to Overlay. We have an option to increase the dot size. So I can increase that dot size to something where I can see it a little bit clearer. And then we can also now see this little FOV gate is showing up here, all red. So as I move this around, that's giving me a little live update as well. Maybe I want to draw a gate in this plot. Super simple. Go to Gate, let's pick the type of gate that I want. Draw it in here. I can rename this. So what's this? This is maybe some small dim cyto cells. And now I chose blue. So all of the cells in this gate are showing up blue in both of my picture plots. So this is what we mean by, you can confirm in your picture the type of things that you're seeing in the rest of your analysis. This updates as I move around. I'm seeing just those cells that the gate is covering, showing me. Maybe I want this a little bit tighter in here. Now we've formatted a few things. So, where we've been changing how we like dot sizes to look. For example, we could go and format the outlay. If we maybe didn't like this brown, we wanted it to look all blue around the edge, something like that. You can come in here and you can make all these formatting choices. And if you want to keep them, if you want every time I make a plot, I want these format choices to be remembered, you can right click and set as default. What this is going to do is, every time you open FCS Express under your login, it's going to remember who the person is and how they like their software to behave. If you want to explicitly see all of the user options that we're applying, they are available to you. They're in File, Options, and then you get this user options window popping up. You can come in here and see the plots and see kind of what user options you've defined for your plots. You can set them from here as well. If you wanted to apply those changes in here, you can. So you have things like how your plots behave, how your statistics behave. We're going to come to this one later. And also, one that's quite important for image cytometry is this data loading. What this is whenever you bring in a file type, we're looking to understand how you like that file type to behave. For example, when you are bringing in, SciPix ACS files, do you want to load the images with them? You can make decisions like that, and we will remember how you like your data to behave. So in the spirit of building up a kind of final presentation, we can come in here and insert some other objects. You notice we have this gate navigator. What this isn't is it's not currently on our layout. It's not printable. If we want that, we can come to gate view. We could bring this in here, and now we have something which is printable and going to be included in our final result. A note on the difference kind of between white and gray space. Everything that is on this white space is going to be included in your output, in your PDF, in your PowerPoint, whatever your kind of output is. If you have any, but you can still utilize the gray space for kind of your own understanding. For example, you can have things off over on the side that are for you. Could even be a plot over in the gray space. That's kind of just for your understanding, but you don't want it to be included in your export. We can also insert a text box, include something like, a title or a summary or any information that we want to, or, you can start to build up these kind of reports. You can also, insert a table into these text boxes if you want to kind of have an output of your data as well. One tool that's really useful with image cytometry data is the data grid. So let's draw one now. So a data grid is basically for every cell or every event, it shows you every single kind of recording that we have for it. So this is a lot of information, right? No doubt. There's lots of parameters here, and there's lots of events here. But you can format it to make it, show exactly what you want. And how do we do that? We come to format, and maybe we want to show parameters to display. So at the moment, parameters to display is showing all items, so every single thing that the file has to offer us. But we can go in here and only show the checked items below. So maybe we just want a little bit of a simplified list, just want to have a look at the cytoplasm and the DNA, and we want to show those images down here. So now we've kind of got this like more simplified list looking at our kind of data grid. If these are now also the images that were segmented for you in your file. If you like any of them and you want them kind of included somewhere else in your presentation, you can just drag and drop them out and use them as you like if you're kind of building up a figure in some way. Something which might be useful to you is the gate information. So what we can do is we can come to here, gates to display. At the moment, we're not telling you any gate information about these cells, but I can hit all gates or a subset, pick out just one or two of them. And then you can tell every cell which gate does it belong to. And you can see this live updates, this data grid live updates with our gates to kind of include them or not include them based on the information. We can apply a gate to this data grid in the same way that we would any plot. You go to gating and you tell it, at the moment, it's a no gate is applying, so it's showing all cells or all events. We could come in here, we could just look at the FOV gate, and this would update. As we move this around, it would just update for that. You can, of course, also drag and drop on here or interact with this data grid as you would any plot. So you can kind of look at this information and start to summarize your data a little bit. Let's put this over the side for us. Another summary tool which you might like to use for your images could be a spreadsheet. Let's add a little spreadsheet in here. So maybe there are specific statistics you want to get out of it. Maybe we want to know the FOV number of events. How many events are in this gate up here? And we have this small dim cyto gate. We just want to know the percentage of all. So we want to put these kind of two events in here. What we can do is all we need to do is kind of drag and drop this gate down here and pop it into our spreadsheet. We'll then get this little create statistics dialog, and we can go through and find the statistic that we want. There's lots of different options in here, but for us, I want the number of events, the count in here, pop that in here. And then rather than, me having just typed in 32, this is now live updating, right? So I move this and it's going to update that statistic live. Same thing, if I want to get this small dim cyto gate in here, drag and drop, go and find the statistic that I want, percentage of gated cells from plot. Again, rather than me typing in 35.43, this is live updating. I can now use this spreadsheet. If I'd added multiple statistics potentially, I could use this spreadsheet to go and make different charts or different plots based on it. So at the moment, something to note about our data, we have two time points. So we have like a 5 and a 10-minute time point, and we have two versions of the images made at those times, a cell and a nuclei version of the image. So what I can do is I can flip between these iterations, and I can see for the two different time points, I can go and I can look at this data. All of these gates are being applied globally, but I could go, and if I was to then go and look at the nuclei images, I could look at those as well. But maybe I want to pair these up a little bit. Maybe I want to look at the two different versions of 5 minutes together, for example. So what I could do for that is, we could drag in this 5 minutes and draw a picture plot, and then I could come in and drag in these 5 minutes and do the picture plot as well. But this changes a little bit how the iterations work, and it changes a little bit how the outputs work and how things like overlaying of different files work. So what I think is a really nice tool for something like this is to come to data and make a panel. What a panel means, it basically just means how should I treat two files or two tubes in a way that kind of is consistent across your data files. So what I can do in here is I can come in here, make a little example, and what I want two tubes. I want to put my two 5-minute outputs together, and I could label that if I wanted, 5 minutes, 10 minutes, or I could maybe I want to put my two cells together and my two nuclei together, however I want this to behave. I could do this, I can apply this, and now it's kind of rearranged my data so that I have two iterations. And if I didn't like the way things were, say I wanted that two 5 minutes together, I can just put them in here, and it's going to just kind of organize things a little bit for me. So that can be a really useful tool for kind of organizing your data, particularly if you're doing kind of multiple kind of files at a certain time point that you want to all reference together. What we're going to do is we're going to have a look at a couple of other application examples, a few other examples of some imaging now. Just going to open some other layouts for us. So we're going to move on to this layout now. For this layout, we've just introduced a picture. These are images of culture plates that have colonies growing on them. We've created some gates here. We've created some against colony area where we have kind of small, medium, and large colonies down here. We also have looked at this red parameter, and we've been able to make kind of white and red colonies. And on the plates, we've actually been able to just draw a gate on two colonies of interest, this kind of big colony 1, big colony 2. And you can see we have all of this kind of information in our gate view here. We've inserted a data grid, which has those images as well as some other information in it. And you can now see the kind of individual shapes and colors of all the colonies. If you are a microbiologist or you're into bacteriology, you know that the size, color, and shapes of colonies all mean something specific about a different strain of bacteria. So we're able to quantitate some of this data in FCS Express rather than just making qualitative assessments. We can see how everything updates in real time. If I was to move these colonies around, I can see the data grid is updating, and I can look at it like that. We can also, of course, if we wanted to, take these images out and include them somewhere else if we wanted to. Drawing on this image, as is like anything, these colonies have all been kind of segmented, so I'm able to go and draw a gate on a particular colony if I wanted to drill down some information on it. So we have all of this information on here. If I was to resize, look at some colonies, we see everything is updating live. So that's kind of, don't think a little bit more broadly. There's lots of different ways you can apply this, FCS Express imaging tools to different analysis types. So let's jump to another application example now. One moment. So in this layout, we're using AMNIS data files or the DAF data file format, and we have a histogram. here, which looks at a single color parameter versus cell number. We've also drawn some nice plots where we're kind of gating out different populations, and they've been formatted in a way which was pleasing. What I'm going to do is I'm going to insert this data list so we can have a look at the data files that we've got. What we have is we have 6 data files here, and they're all taken across different time points. So we have a 0 hour, 1.5 hour, 3 hour, 6 hour, 12 hour, 24 hour. And we can move through these data files and we can look at how the kind of analysis is changing as we go, be that in the plots or be that in the data grid or in this overlay. But maybe what we want to do is we want to compare kind of all of these samples back to the 0 hour and see how things are changing over time. What I can do is I've got this 1.5 hour one selected. I can then drag the 0 hour on top of this overlay and tell it to add the files to the plot as a new overlay. We've now got that 2 file in here, but the formatting is maybe not exactly as we'd like. So we can come in here and we can format this overlay. Now we can see there's lots of different ways that you can customize your histograms. We can see that this histogram of our 1.5 hours has been nicely formatted. So let's go to the 0 hour and maybe do a similar job here. Maybe we want to fill the histogram area. We want to change the color. Maybe we want to increase that transparency a bit. And maybe we want to decrease the smoothing to make it a little bit more similar to the previous one. So now we have this nice overlay where we can kind of see the differences between our 0 hour and our 1.5 hour. But if we were to start going through the other iterations to kind of maybe look at 3 hour or 6 hour, we won't be comparing back to 0 hour. Instead, we'll be just comparing one file back. How do you avoid that? Well, what you can do is you can come to this 0 hour and say, this overlay, I don't want you to change when you move when you go through different files, when you do next or previous or select a different iteration, always keep this 0 hour. And now, if I was to click through to look at 3 hour, you'll see 0 hour against 3 hour. If I click to look at 6 hour, you'll see 6 hour against 0 hour. So I'm able to now have this kind of overlay looking at different populations as I go through, and all of these iterations are now comparing the information that I want them to. So maybe once you've done like analysis like this, you might think, okay, I want to get some statistics out, and maybe I want to get, get a slide out for this information or a PDF out for this information. How do I do that? Well, that's done via batch actions. Batch actions in FCS Express are essentially every time I use a layout, I want you to do something for me. I want you to do an action. These are a little bit like macros, where you'll just set them up once and hit run, and they'll always happen every time you use the layout. An example could be to save this data to PowerPoint. Another example could be to export to Excel, something like that. Let's go through a couple of examples here so you can see exactly what the process looks like. So for save to PowerPoint, you're going to get a few options of exactly what you want to happen. I always like to click open the presentation after saving. So, I don't have to go and find the PowerPoint. It just opens up for me to have a look. You can change the page that you're looking at. This is just a one-page layout, but remember before we had multiple pages, so you can select those pages or just use the current page that you're looking at. This iterations options, I think, is worth spending a second on to talk you through what the top 2 options mean. So you have saved the output of each iteration to a separate presentation. In our example, one iteration is one file, and each iteration has one page. So that would mean that each iteration has its own presentation. So we would have 6 separate PowerPoints, each with one file and one page in it. In the previous example from the last layout, where we had 2 files to an iteration, it would carry on that behavior based on iterations rather than the file types. You can also combine the outputs from all iterations into the same presentation. And in this way, in our example, with our 6 iterations for 6 files, you would have one PowerPoint with 6 pages, so 1 page per file type, essentially. If you are if you're doing like a longitudinal study where maybe you are these six files are part of a longer study and you want to just add them onto the end of a previous PowerPoint, you can do that. You can just append it onto an existing PowerPoint. Or if this is the first time you're using this layout, go and save to a new file. You just come in here, click this yellow folder. Let's go and find, let's set a quick name. Come in here. I always think it's worth asking for confirmation before overwriting an existing file. What this means is basically, if you're going to be using the same layout multiple times, running batch actions, before you overwrite any of your previous saves, you'll get a little pop-up telling you what's about to happen. So now we have that as an action. If I was to hit run, I would make that PowerPoint. Let's add something else. Let's add an Excel. So we have a few little options in here. Export to spreadsheet is exporting internally into an FCS Express spreadsheet, whereas export to Excel column mode is exporting to an Excel outside of FCS Express. And what column mode means is that in your Excel, you're going to have a row is going to be an iteration. So in our example here, we would have 6 rows for our Excel, and then we would have as many columns as statistics we add. So it would be iterations as rows, statistics as columns. So let's go and add one of those. Again, we get the same kind of options of, do you want to append it to a previous Excel, or do you want to add it to a new Excel? So let's go to our desktop and go. Oh. And add that in there. We can also ask for confirmation before overwriting if we're running multiple things. Now, if I was to hit run, we would make an empty Excel because we haven't told it any statistics to add. So how do we add statistics to our batch actions? Exactly the same way we added them to the spreadsheet before, just with a drag and drop. So let's say I want these, the percentage of these 3 gates in an Excel. I could drag over here, choose the statistic that I want, percentage of gated cells from plot, drag to my Excel, find the statistic that I want, percentage of gated cells from plot, and do that for all 3. So now we're going to have an Excel with kind of 6 rows and 3 columns. What won't be super explicit is exactly which file belongs to each row. It will be in the order that we have here, but what I think can be really nice to make this super explicit is to add one more statistic. That is, what file name is each row representing? All of these plots and gates have all come from the same file name. So in our example, it wouldn't matter which one you pick, but what you can do is you can just drag and drop one more statistic and find this file name. And this is going to really help you if you're kind of coming back to revisit this Excel later. It's going to make it very explicit exactly what that Excel is about. At the moment, it would be the 4th column. Maybe you want it to be the 1st column, so you can just reorder things. What you can't do is you can't just directly drag and drop up. You can see that little no sign. You kind of shift to the left, see this blue line, and then you can reorder things. Something else which can be important for you is, what are the columns going to be named? So what is the top of this statistic column going to be named? We have some default behaviors in FCS Express, which we're going to go look at in a second. But if you want to be super explicit and you want to call the column something exactly as you want it, like maybe you would in this table here, you can just double click this statistic, go to field, then you get this free text thing. So you can come in here and you can name a statistic exactly as you like it. If you're reusing a layout for kind of multiple times, it can be worth spending the time getting these things exactly as you like them. However, there is some default behavior. I'm going to show you what that is. It's in your user options, right? So remember, user options was file options. And if we go to statistics, we have statistics naming options. So here you can kind of change how those statistics are going to be named. If this is your first time using FCS Express, I'd really recommend spending 5-10 minutes exporting some statistics, having a look at what they are, coming in here, changing things around, exporting, having another look. And you kind of understand exactly how you like your statistics to be named. Change things like, is the population going to be at the beginning or the end of the statistic? change things around and see which one works for you. So now, if I wanted to run those batch actions, I can. But what we do for batch actions is we start wherever you're selected and just go down the list. So we're actually looking at sample 6 here. Maybe it's a little bit more obvious if I open iteration 5. What we'd do is we would run the batch actions for iteration 5, then run the batch actions for iteration 6, and then stop. We don't loop back around to the top. This is because we mentioned those kind of longitudinal studies where maybe you're adding kind of multiple files to the same layout over time. You're just going to want to run your batch actions on your new files, right? So you would select, say we had done this on 4, then added 2 more. You just come to 5 and run the 2 new ones. If, however, you want everything, it's the first time you're using this layout, just make sure you select the top iteration. And then when you hit run, it's going to go down this list nicely for us. So let's hit run now. Just going through those files one at a time and making that for us. We're now going to open our Excel, and we've opened a PowerPoint off screen. So let me bring that in for you guys. So we can see here, we now have this PowerPoint where all 6 iterations are in one slide. We can come in here if we wanted to, edit this. So we could have a look at all of these different things in context if there was something you wanted to edit in your PowerPoint. And we also have this Excel. Notice we have this first column, file name, super explicit, how things are organized. And then we have these statistics again down here. So these are the type of things that I talked about. Maybe you want a free text this name, or maybe you want to edit in your user options. Up to you to get exactly the statistic that you want. So we're just going to open one more layout for us today. So this is just another way that you can visualize your image data. Here we're using some cell profiler data. You can see it's a .cpout file in our data list. And we've inserted a couple of plate heat maps. So you can see data for each of the wells in a 96 well plate, and we can compare them side by side, and we can immediately start to see the difference in our treatment conditions. On the bottom, this heat map is linked to this gate. So as I move this gate around, you can see that I'm live updating my heat map. We can edit this heat map in the same way that we've edited everything else. We can come in here and edit the format. So to do that here, let's just bring that over. So in this overlay section, I can pick the parameter that my heat map is showing. I can edit the statistic that I'm showing. Another tool which can be useful is a different type of visualization where you can change the radius parameter, right? We could change the radius to size based on a parameter, for example, the area here. So you can just get another bit of information out of your heat map. That can be really nice to see. Come in here. We can also change the color, the coloring statistic in here. So at the moment, we're using flame just based on the percentile. I can change this if I wanted to maybe look at a different color format. If I don't like this kind of fixed range or percentile, I can also use another option. I'm just gonna come and turn that radius parameter off. Let's go to color levels. Something which is nice to do is to use threshold. And then what you can do is you can just select kind of 2 colors for kind of an on-off presentation. So say we just wanted to know if something was above 150, tell me. Here you can kind of scan all of your data really, really easily, and you just get to know on-off. And maybe kind of these black wells are the ones which we would take forward, were of interest to us to kind of dive a little bit more deeply into. And you can see all of this kind of live updates based on whatever you're looking at. So this can be a really nice way if you're kind of just scanning data to kind of get a quick answer, kind of putting these kind of thresholds in. This top plate map here is actually showing a couple of things. What we've done is we've overlaid 2 different statistics in our heat map. So for each well, we're looking at this cytoplasm output and this DNA output. And you can see they can be edited or changed. You could add a radius parameter to get kind of a third thing going on as well. And you could also come and change the color levels as well. You can edit all of those things. And then you can see we're able on this other page just to bring together all of the kind of things that we've looked at today. We're able to come in here and move these gates and the data grid updates, the picture plot updates, and we can kind of see things going on as they do. So let's come back to our first presentation. So just to go over some of the highlights of FCS Express that we've seen today. FCS Express has direct compatibility with multiple file formats, the ICE format, the NXDAT format, DAF, the cell profiler output formats as well. It can be used to work directly between raw data, plots, and images to produce high quality data visualization, plots, and reports. We have data analysis tools for highly multi-parametric data. We didn't really talk about this today other than showing you where the presentation was, but FCS Express can perform a number of data transformations such as TISNI, VISNI, cluster analysis, PCA, SPADE. If you have something very specific working in your institute in R, you can also integrate R with FCS Express to run that with us as well. FCS Express offers unique features for advanced analysis and reporting, such as batch processing, overlays for histograms and plots, and integrated spreadsheets. There are also algorithms available for techniques like cell cycle modeling and regression analysis. So you don't have to move to another software to do that type of analysis. You can do it with FCS Express. If you haven't tried out FCS Express yet, we do offer a free 30-day trial of our software. It's fully functional, so you can try everything you've seen today and everything we have on offer and see how it works. You can download it from our website, denovosoftware.com. And if you have any questions that have come up today during our presentation or anything at all you want to know about FCS Express, please write into support. That's support@denovosoftware.com, and we'll try to get back to you either over an email, or if it's something you want to set up a training for, we're happy to jump on a call with you. So I'm just going to point you at a couple of resources now. So we saw that image compatibility table, so where we can see, exactly, and if you're watching this, a few months after this talk, you can come in here and have a look. Have we added any new products or file formats to our image cytometry output? And also this applications example. So I mentioned if you want to get a start, you can go through and have a look at the tutorial, but we also have these applications examples. This is where we've gone through and a few different instruments or types of analysis. You can come through this and have a look at how we'd maybe set up a layout or how we'd go about analyzing things. If your type of analysis isn't here, please email us, support@denovosoftware.com, and we're happy to help you get started, so you can help get started building a layout for your analysis. So thank you for attending today, this webinar, Getting Started with FCS Express 7 Image Cytometry. Please feel free to reach out to us, support@denovosoftware.com, with any questions that have come up today or any questions you have about the software whatsoever. And please visit denovosoftware.com for any additional webinars or any videos that we have or any of those articles to help your journey in getting started with FCS Express. Thank you all. Thanks for attending, everyone.